First, um, hello everyone, once again. So I am taking you to the Blackboard page. So uh, you have the syllabus yesterday, or sorry, yesterday means on Tuesday. Based on our discussion, I updated the syllabus, and this is a recording of the syllabus. And I will be posting as we move along. As we finish the chapter and we move along. I'll be posting each chapter as a learning activity, and you can click this. You have the teaching slides, you have the assignment. This is the discussion board one. It has assignment and discussion board. Board is also a type of assignment. So it has a deadline, so check the deadline, and similarly the assignment. And after the class, I'll be posting the recording. So this is like a typical learning activity that will happen across the semester. And for example, this chapter can go for, it's a big chapter, so we can split this into, this could be week one or day one, and then the coming Tuesday will be, we'll finish this and we'll have the assignment, and I will put the date according to when I finish the chapter. Okay. So you have to keep on top of the Blackboard. So chapter one is the development of molecular biotechnology. So yesterday means last class, we discussed at a broad length about some of the key discoveries and how they connect to modern day problem challenges, and where we have hope, right, to fight those or to take care of those challenges. So this is the first slide. The term biotechnology was introduced in 1970 by Hungarian engineer, by a Hungarian engineer, and it's hard to say molecular biotechnology, how much it will cover. A molecular biotechnology will include, of course, the functions of DNA. For example, DNA functions as a template for replication, template for transcription that leads to translation. And even if you talk about RNA, that's also part of molecular biotechnology, proteins and carbohydrate, lipids. They're all molecules or macromolecules. They are the building block for our cellular structure and functions. And as much as we know, there are many things unknown. So you are studying this course as a student of biology to strengthen your knowledge, so that if you want to have a career in biology or in healthcare profession, then these will go a long way. And one of the major discovery that laid the foundation for molecular biology was the structure of DNA by Watson Crick. Before that, in 1953, before that, the field was mainly called as genetics, right? People used to look at the chromosomal level, right? And how they looked at chromosomal level, they descended from the Mendelian genetics or Mendelian principles. Why are called Mendelian? Mendelian means Gregor Mendel, because Mendel in his principles, in his theory, he talks about character. He doesn't have any knowledge of chromosome, DNA, nucleotide, so and so forth. So he refers them as characters. So for example, color of the flower or height of the flower or the pattern of the leaf. Mendel was very hardworking, very analytical, very systematic, but he was also very lucky also. He was lucky that if you go back and recall your knowledge of genetics, there are various characters, height, color, shape of the leaf. All these genes were located on the chromosome at far distance. What do I mean by that? Is that let's say if this is the chromosome, right? If this is the chromosome, then Mendel was lucky that the genes were located not nearby, right? Not really nearby, but they were located at a distance. I'm telling you for your understanding. This distance, right? This this distance prevented a lot of complexity, right? And if Mendel would have encountered this problem, then perhaps he would not have been able to propose his laws that became law, but his observation, his theory, right? So these characters were located, little bit farther on the chromosome, so he could propose discrete law. But that is not the case. Some genes are located very, very, close, very, very close to the with each other. So they inherit together, and the effect is cumulative. So what Mendel was talking about, the character, was actually in the modern world, in today's world, is genes. So what are genes? So genes are the segment of DNA that can code for RNA, or this RNA can go for translation. So that's a gene that has a capacity to produce RNA or express RNA and express protein, and they have a function. So that was the starting point when scientists thought that what character Mendel is talking about. So then from Mendelian principles to it went to genetics, because people could figure out that this is chromosome that he's talking about. And on the chromosomes, there are genes. But until 1953, uh, people did not know the structure of DNA. And what is most interesting for the DNA structure of DNA was people, Watson and Crick, could figure out that DNA is double-stranded, made up of nucleotides, but they did not knew how these two strands are connected. So Watson and Crick, they knew, or many people knew, including them, that DNA is double helix, but they did not know how this is connected. So I'm coming back to Mendel. I'm talking about Watson and Crick. So they visited uh, Rosalind Franklin laboratory in UK, Cambridge, and there they got some insight into the X-ray crystallography data, and there they figured out that these two strands, they are linked by hydrogen bond, and they got the Nobel Prize. But sooner people realized that that Watson and Crick, they had no experimental clue for the hydrogen bond. It was the pivotal work of Rosalind Franklin that helped Watson and Crick to to um, to discover DNA. So so it became very clear that maybe the Nobel Prize would have been awarded to all three of them. But regardless, we are talking about So that was a major discovery for DNA, because what I was telling you yesterday in our last class, that we are in the era of genome editing, where we want to correct the mistakes that happens in our DNA, that would take care of many genetic born diseases and illness. Um, so then one of the ways to manipulate DNA is to use restriction enzymes, right? Without restriction enzyme, uh, it would not have been possible to. It's not possible to manipulate DNA. So I see that people are still coming in, so I'm a little bit distracted. So let me just get people in. Yeah. Yeah. So, I will, we have to live with this. People are coming, a little bit late. We are 18, 19 minutes, 20 minutes into the class. And so I have to pause. So my apologies, because I'm recording, but I have no choice. And then in late 60s or early 70s, Hargobind Khorana, his work on RNA, he could crack the significance of genetic code. And one thing I would highlight the importance of genetic code is that whether we talk about viruses, whether we talk about bacteriophages, and whether we talk about DNA viruses or whether we talk about RNA viruses, talk about plants, we talk about plants. Uh, so we talk about plants. Uh, you know, it's all DNA, but there is a world of RNA also, right? So genetic material could vary in species depending upon virus, plants, animals, but genetic code is preserved. Whatever the species could be, virus, bacteriophage, DNA virus, RNA virus, the genetic code it stands alone. There is no exception to genetic code, and that's a major discovery that I think that happened in the field of molecular biology. If that was little bit, uh, confusing or foggy or blurred, then our concept will not have been so solid. If the genetic code, especially the discovery of the stop codons and start codon, that's really a very, very fascinating work. Then animal vaccines using recombinant DNA technology. We'll talk about this later, and I will talk about in with regards to production of insulin at the industrial scale. Then the discovery of PCR was very important. Why it was important? Because for the first time, human or scientist got the capability to amplify genes. So, so scientists could amplify. So let's say there's one copy of gene, right? And then scientists got that strength, the tools to amplify this one copy of gene into more than 1 million copies, right? One more than um, millions of million copies in few hours, right? In few hours, and that led to a major shift in the field of diagnosis, right? Because it increases the power. Before that, people used to do mainly pathology. They used to look at the values, and they used to take, the labs used to take 1 hour, 2 days, or 1 week for to get back to the doctor to start the therapy. But PCR, especially in infectious diseases and cancers, and PCR was very effective to find out what kind of mutation, what kind of infection is there. For example, when uh, I was doing my PhD, PCR was very instrumental in finding out quickly whether a patient is HIV positive or negative in few hours to confirm at genetic level. Um, then then RT-PCR was developed, and then this RT-PCR was able to detect viral load in a patient, meaning how much amount of HIV is circulating in the blood, and um, this would determine um, what what kind of uh, what kind of treatment should be instituted in these patients. If the viral load is high, then different aggressive treatment, and so on and so forth. So PCR was a big deal in the field of molecular biology and molecular diagnostics. So so this is the few very important, and then of course human genome sequencing. So human genome sequencing helps us to know which gene is where, what uh, kind of sequence is there, what are the adjacent genes, and then what is happening in variants, mutation. So there's a library that we can access to compare the genome, and it will have far consequences at as slowly, particularly people are very interested now in evolution. How did you know at uh, what point you know at what point humans diverged in evolution, and what was happening in Europe or in certain parts of the world that we will look at some different day. So these were some of the major discoveries that happened in the field of end of biology, and the discoveries continuing to happen. And I mentioned two names. One is CRISPR technology that is being used for genome editing, and second now is the mRNA vaccine platform that is being used to produce COVID-19 vaccines. So this is the list of many discoveries that happened, significant discoveries that happened in the field of molecular biology. So this is just given till 2009, but I'm sure the newer edition there will be more. Now, so as I mentioned to you just now, I was reading the news in last 1 or 2 days that Joe Biden, the president, is taking initiative to reduce the cancer by 25, by significant, I don't remember the number, by reduced by 25%, or to treat cancers in 25 years. So I'm confused. I'll get back to you. But there's a recent announcement by Joe Biden for expediting the cancer treatment or cancer research. So genes are important, and genes are functional unit of producing right protein at the right time. And if the gene is mutated, then the protein is problematic, and the cellular function is compromised, and that gives rise to diseases. And so these mutation can occur in a regular way, or people can get mutation from birth. So there can be two kinds of mutation that can happen, either by inheritance or by exposure to environmental agents or certain kind of stress. And another fascinating discovery that is, trials that are going on, or effort that is going on, is to is to treat in those individuals who are carrying genes which are not which not have healthy babies. And particularly one of the things that was not studied in deep was mitochondria, right? Mitochondria is very important. Mitochondria has its own DNA, and mitochondria is inherited from mother rather than father, right? So if mitochondria is not good for any reason, then there are strategies to replace the healthy replace the sick mitochondria with healthy mitochondria. So this is unhealthy mitochondria, and this is a cell with this is unhealthy. The bluish color and the green color is healthy mitochondria. So the nucleus, the pronuclei, one from father, one from mother, can be extracted, and then it can be uh, placed into cells or embryonic cells or stem cell with a healthy mitochondria, right? And so these kind of reconstructed embryo, right? So this kind of effort has been going on to to minimize uh, you know, genetic disorders. Now I'm a little bit switching gear from history and development to what are the systems, what are the model systems that are being used for discovering or for to developing um, molecular biotechnology tools, right? And if you have the right tools, then you can put in place a kind of a platform that can be used for benefiting individuals or society in many ways, right? And so one of the most important organism, because we have to understand that human body cannot be used for research because of ethical reason, and also because of slow results, right? So if you're looking for changes in genetics, then one has to wait for 9 months, and then just so it's a long time taking. But if we develop a model system in bacteria, fungi or yeast, which divides very faster, then one can see biological phenomena in these model organisms, and from there they can conclude or they can speculate the function of a gene. Similarly, insects. So bacteria is a prokaryotic organism. Yeast or fungi is unicellular eukaryotes. And insects are multicellular organisms, and plants are they have chloroplast, chlorophyll, so they're different part of the kingdom. But slowly and slowly, scientists, they are using human or mammalian cell line, and because they want to get to a model that can resemble a disease, or it could be very close to human settings, right? But still, these are not very efficient models, but we get the results from which we get an idea about a gene or a protein. So I am sure you know the differences between eukaryotic and prokaryotic cells, and that these differences, it is because of these differences is because of number one, nucleus. In bacteria, there is no nucleus. The DNA is directly in the cytoplasm, right? And this is protected by cell membrane and cell wall. So there is no well-formed nucleus. So this DNA and protein complex is referred as a nucleoid, right? And in humans or in eukaryotic cells, there's a well-formed nucleus. And within the nucleus, there are chromosomes, right? And also organelles. Bacteria has ribosomes. They have complex cell membrane and cell wall. But in human cells, they have mainly plasma membrane, which is semi-permeable. And there is not only ribosome, but mitochondria, endoplasmic reticulum, Golgi apparatus. So a mammalian cell or eukaryotic cell is full of specialized organelles, right? And so on, so forth. So a bottom line is that that there are various model systems that can be used to understand the diseases, human diseases, or to develop some therapeutic molecule that can be used for treatment. So these are some of the microorganisms that can be used for growing and for making molecules. For example, E. coli. E. coli is best suited for recombinant DNA technology. Then Erwinia herbicola could be used to make vitamin C. Rhizobium, I'm sorry, for nitrogen fixation. They can be genetically modified to increase the plant, the nitrogen cycle in plants, so and so forth. So the most important thing is that when we are working with bacteria, it is essential that bacteria are not pathogenic, right? So like it doesn't cause infection in the lab. So these bacteria have to be dealt in a or have to work to be in a very very septic or in a very very protected safety conditions. For example, this is not a bacteria, and most people get confused. This is single cell eukaryotes, and they are non-pathogenic single cell yeast. They convert sugar into ethanol and CO carbon dioxide. But this yeast S. cerevisiae can be used for fermentation, and they can make protein. Now, one of the most challenging thing that happens when we use bacteria to make human protein is that, so let's say if so let's say this is a bacteria, right? And bacteria has, let's say, DNA, and bacteria makes RNA transcription, and then they translate into protein, right? Bacterial world protein, 3D structure. So DNA transcription and then RNA into translation with the help of ribosome. Now, this bacterial system is very simple, but if you look in the eukaryotic cells, there is a nucleus, right? This is a nucleus, and this nucleus houses the DNA. This DNA can be transcribed into RNA, and then this can be further translated into protein. Right? But what I am telling you here that you can pay attention is that that human proteins or mammalian proteins can undergo chemical modifications with the help of enzyme. These modifications that occur on the protein is called as post-translational modification. And there are a battery of modification that can happen in a eukaryotic cell, right? So one thing to be very precise, please don't get confused, that that I'm realizing now is that, just not to confuse you, that right that that in the nucleus, this is the nucleus, then this is the DNA. This DNA then transcribes into RNA, right? And this RNA goes out into the cytoplasm. In the cytoplasm, this RNA, just to be very precise, just in the cytoplasm, this RNA translates into protein, and this protein in eukaryotes can get modified chemically with the help of enzymes, and these are called as post-translational modification. This is very different from bacteria. Bacteria generally don't have this kind of array of post-translational modification. So when you are making a human protein in bacteria, they don't have the same enzymes that does post-translational modification. So that's one of the challenging aspects to work with bacteria. So that's why, so I repeat, bacteria have less enzymes like humans that can post-translationally modify proteins. So it is always wise that if you need a protein for therapeutic purpose or for functional purpose, then it is good idea to express or to produce the protein in yeast, because yeast is a eukaryote. So they have those enzymes, those battery of enzymes that can be used to modify proteins. And secondly, in presence of various chemicals, yeast can be used for fermentation, and this fermentation can scale up the production of proteins. So just again, this is the central dogma of biology, that DNA is a genetic material that transcribes into RNA, serves as a template for transcribing RNA, and the relationship is complementarity. Means if the DNA has A, RNA will have U. If the DNA has T, then RNA will have A. If the DNA has G, RNA will have C. And if the DNA has C, RNA will have G. So there's a complementarity relationship between DNA and new RNA, right? And this RNA can be made into, or this RNA can serve as a template to make proteins, right? So this is the straight path. But what is the exception here? Exceptions are the viruses, which are very, very mysterious. And we are dealing with one where RNA can be the genetic material, and this RNA can be either transcribed into DNA. RNA, remember, I'm just saying, listen to my word, RNA can be transcribed into DNA. But such transcription from RNA to DNA is called as reverse transcription. DNA to RNA is forward transcription, but it is referred as transcription. From RNA to DNA, it is called as reverse transcription. This is also mediated by the enzyme called as reverse transcriptase, which are present in retroviruses such as HIV. Now, COVID type of viruses, they also have RNA material as genetic material, but they don't have reverse transcriptase like HIV. They have RNA dependent RNA polymerase. So remember when the pandemic started, there was a huge big deal about remdesivir, and so that remdesivir could be therapeutic, but it was not. But the target for remdesivir was RNA-dependent RNA polymerase in COVID-19. But somehow that medicine was, I think it was first generation, that was not much effective in case of COVID. It was not like, it might be effective in subset of patient, but it was not like ubiquitously effective. It could be complicated by the stage and the complication of the disease. Some people got it and some people got better, but not everyone. Stage of infection, stage of symptoms, and many complex things. So RNA viruses could have either reverse transcriptase or RNA-dependent RNA polymerase. And both these proteins, reverse transcriptase and RNA dependent RNA polymerase. They're the target of modern biotechnology company to take care of the disease. But just recently, there was also one new strategy has been developed for the treatment of HIV. I will cover this as and when we keep moving forward. So I mentioned to you that I will also take this opportunity to just clean up the bases or basic knowledge, or so we all know that the component of DNA. Now, why I'm telling you this is that you have to remember and you have to recall this concept when we are talking about sequencing or DNA synthesis. So DNA is a polymer, a macromolecule made up of a polynucleotide. One single nucleotide is made up of a sugar, uh, phosphate, and nitrogenous bases. And they are of two kinds, purines and pyrimidines. Purines are bigger molecule because they have double ring, adenine and guanine, uh, and cytosine and uracil, right? They are single ring is small, and the base pairing is between purine and pyrimidine, A with T and G with C, right? Now, what I want you to emphasize here, that don't look at these as just as a molecule, but you have to orient your thinking of the excellent chemical engineering that goes inside our body in making this nucleotide, right? This whole chemical thing is generated inside our cells, right? After metabolism, right? And so our body is nothing but chemical factory, right? Making this complex molecule, which is very extremely difficult to synthesize outside in the chemistry lab. Now, more sophisticated is the orientation. What do I mean by orientation? Is that 5 dash, 3 dash, right? So remember I spoke yesterday that this is a sugar molecule, which is ribose, and ribose have 5 carbon atom, 1, 2, 3, 4, and 5th carbon atom. Now, out of which, 3rd carbon atom is most sterically friendly for the synthesis of DNA template. Sterically more, um, easy to accessible sterically. Steric means, in a three-dimensional conformation, sterically accessible to enzymes such as DNA polymerase. Now, this phosphate molecule, which is very, important, because this is the phosphate molecule that gives negative charge to the DNA. Had the DNA been positively charged, the story would have been totally different. The DNA is negatively charged, helps the DNA to be solubilized in our nucleus, right? So otherwise DNA would not have been soluble, or the pH of the cytosol could have been different, something. So, but look at this phosphate molecule, right? So this is the central phosphorus atom, 1 oxygen, 2 oxygen, 3rd oxygen, which is negatively charged, and the 4th oxygen is a bridge between the phosphorus and the 5th carbon atom. Now, this is very complex chemistry, right? But this happens in seconds in our body, right? So, you have to ask yourself question that how come this bonding is so specific happening? Why not it happens here? They are all oxygen, but why in this direction? So that these things we will cover in as we go forward. Now, phosphodiester bond, right? As I mentioned to you, between 3rd carbon atom and the 5th carbon atom. And so the starting is 5th, 5 prime, and then 3 prime. 3 prime is most sterically convenient to make phosphodiester bond with the 5th carbon atom, right? And also, I'm sure you know that in DNA has deoxyribose, right? So the sugar has 1 less oxygen, and this oxygen is lacking on the 2nd carbon atom of the ribose molecule, right? then here growing strands, right? It grows in uh, in um, in 5 to 3 direction, and the mode of replication is semi-conservative. What is meant by semi-conservative is that uh, let's say if this is the 2 strands of DNA, then semi-conservative means, so these are the 2 parent strand, right? So these are the uh, parent strand, right? But here, each parent strand separates, and then this gives rise to the daughter strand. So half of the DNA is original, half of the DNA is new. Half of the DNA is original, and half of the DNA is copied. What is meant by copying that? It's complementarity. A, T, G, C, C, G, T, A. Right? So half is original from parent DNA, and half is new. So it's called a semi. Semi means half. And and DNA is nothing but a very excellent biomedical engineering, right? So it is so everything is very very calculated. So now here, be careful. The distance between 2 adjacent nucleotide is 0.34 nanometers. I repeat, 0.34 nanometers. But if they write in angstrom, right, it will be 3.4, right? 3.4 angstrom. And DNA, the one turn, right? So DNA is helical molecule. So from this end, going from this here is one turn. And within one turn, there are 10 nucleotides. So 10 multiplied by 3.34 nanometers is, so one turn, the length is 3.4 nanometers, right? So and if you're calculating in angstrom, then you just multiply by 10. And also, as I mentioned to you, the work of Rosalind Franklin was very, very instrumental. Otherwise, no way Watson and Crick could have discovered the structure of DNA. And so number of hydrogen bond is very critical. A and T are bounded by double hydrogen bond, and G and C by triple hydrogen bond. So you should be very, you should pay attention here also that these 3 hydrogen bond, so the energy needed to break hydrogen bond between A and T is more energy required to break. Why do we need to break hydrogen bond? That is the main thing for PCR or for DNA replication or transcription, the separation of 2 DNA strands, meaning breaking of hydrogen bonds. So the energy required to bring 3 hydrogen bonds between G and C is required is larger as compared to double hydrogen bond between A and T. And that was the information that Watson and Crick got from Rosalind Franklin, right? And DNA is anti-parallel. So DNA is parallel molecule, but they are called as anti-parallel because one of the strand runs from 5 prime to 3 prime, and other strand runs from 3 prime to 5 prime. So where does this 3 prime and 5 prime comes from? This 3 prime and 5 prime comes from the sugar molecule, right? Okay. Now here I'm talking about, remember I mentioned to you when I was talking about Hargobind Khurana's work, that he with his collaborator, he could discover the genetic code. And this is all maths, right? Because what does genetic code represent? Genetic code represents the relationship between mRNA and its protein, right? So that's translation. So how do we know that a particular RNA will code for which proteins, right? So it was discovered that the 3 bases of mRNA codes for one protein, one amino acid, right? I repeat, 3 bases of mRNA codes for one amino acid, out of which In many 99% of the cases, the first amino acid is methionine, or it has a code as AUG. There are 3 stop codons here. So out of the 64 triplet, 61 codes for amino acids and 3 codes for stop codon. Stop codon serves as a message for the translation to stop at the surface of mRNA. Now there are 3 kinds of RNAs, right? mRNA, tRNA, and rRNA. All they have their roles in translation, in bringing amino acid to the surface of ribosome. Then rRNA is instrumental, is making peptide bond. Right? And now if you look at the typical gene in bacteria, there's promoter to which RNA polymerase bind, and there is one gene that can make one protein, and this protein can translate it. Right? Or there can be several genes together, adjacent from one end to the other end together. But eukaryotic is complex, right? Our chromosome or our gene is a mosaic of exons and introns. Exons are also part of RNA. Introns are also part of RNA. But the difference is that exons can code for amino acids, introns cannot. So introns have to be taken out, spliced. So there's another machinery called as splicing machinery. So all these part that leads to the removal of introns or capping and tailing, this is called together as RNA processing. So I repeat, eukaryotic gene, which codes for a protein, is a mosaic of exons and introns. After transcription, the introns are removed, and then the exons are connected to each other. And then there is capping at the 5' end of mRNA, and there is tailing of poly A on the 3'. This is called as RNA processing. After this, the RNA travels to the cytoplasm where translation occurs. And there is one more step that I mentioned earlier, is post-translational modification that occurs in mammalian cells. So these are, hopefully, this is what I said. I just mentioned to you in earlier slide that introns are removed and exons, they come and join each other. And then there is a special sequence called as Kozak sequence. This is present on mRNA, and they help in the process of translation. And I mentioned to you a structure called as promoter. So promoter is a site where RNA polymerase binds, and scientists have studied that there are some areas in the promoter which are very conserved. So how I use the word conserved, so how scientists came in with the conclusion that these are conserved area. This was based upon sequencing amongst many species, and they concluded that so one of the conserved area is called as theta box, which is located at -25. Now, what does minus stands for, and what does plus stands for? So any base, so if this is the starting base for transcription, the starting base can be called as +1. Anything before the starting, before, is in minus. -25, -75, -90. So these are the conserved boxes or sequence of nucleotides at -25, means before the starting nucleotide. So these are also called as upstream, before the starting base, it is called as upstream. Anything after starting base, they call it downstream, and they are labeled with positive plus sign. These are before, upstream, with negative sign, minus sign. Then the translational machinery is the ribosome. Ribosome serves as a platform, as a stage on which uh, three RNAs comes. One is mRNA. All these RNAs, they are coming out of the nucleus, and mRNA is sitting on the ribosome. Then the tRNA comes with the first amino acid at the P side, then the second at A side. Then this peptide bond is synthesized, then they move across the base, and the translation occurs, right? And the translation stops at the stop codon. Now, this information is very important this is central to the production of insulin, right? If we don't know the gene of insulin, if we don't know the right mRNA for insulin, if we don't know the right uh, amino acid composition of a insulin hormone, then how can we treat diabetes, right? And how can we make insulin at the factory scale? So we need the knowledge of insulin gene, right? Insulin gene. Also, I want you to think here is that, so we inherit half of our chromosome from our mother, half of our chromosome comes from father, right? So the total number of chromosome is 46 in our somatic cells, and the chromosome is actually two sets of 23 set from mother, 23 set from father. But that's one concept. The second is that uh, each body of our cells, each cells in our body have the same DNA because they're emerging from zygote, right? One zygote is formed after fertilization, and that zygote grows and grows and grows and makes an individual. In 9 months, an individual is delivered. All the cells in our body is coming from zygote, from the mother's body, that is coming from fertilization. So meaning that all the cells contain the same DNA, yet some cells make different proteins. For example, RBC cells, they make hemoglobin. Hemoglobin carries oxygen. Where is RBC formed? They're found in bone marrow, right? But as far as insulin is formed, insulin is expressed in a very specific tissue, that is pancreas. And within pancreas, also in a specific cell type that is called as beta cells. And the alpha cells and beta cells. Beta cells makes functional insulin. So this insulin gene has to be identified, right? So this can be identified and amplified by PCR, polymerase chain reaction, and then this gene can be inserted into a vector. Remember I told you what is the vector? Vector is a device, right? Although it's DNA, it's a device that can carry foreign DNA. These vectors are product of bacteria. Plasmids, they're present in bacteria. So what is bacterial body looks like, or bacterial cell looks like? So let's say this is the bacterial DNA, this is the bacterial RNA, this is a protein. Now, in addition to this, bacteria also has an additional extracellular chromosome. This additional extracellular chromosome, which is also double-stranded. This is double-stranded DNA. Right? Now, this is double-stranded DNA. This is independent of this. So this is the main. This is the main DNA of the bacteria. And this plasmid is the extra chromosomal DNA. Bacteria can survive without the plasmid, but bacteria, they cannot survive without the main chromosomes. They are both independent of each other. They can make separate proteins, but this plasmid can be used as a tool for making recombinant insulin. So the insulin gene is taken, and this is ligated to the bacterial plasmid. And then this bacterial plasmid is inserted back to this bacteria. And as bacteria is growing, the insulin is being produced. So this is the simple concept of recombinant DNA technology. Why it is recombinant? Because, because this vector is coming from bacteria, right? Plasmid. And this gene is coming from humans, insulin. So there are two DNA, right? So you're recombining. So this DNA is recombinant, one from human, one from bacteria. But bacteria body serves as a host, as a lab, as a factory to make excess of insulin. As bacteria is growing, dividing, they are producing more insulin. So from next class, I will start, next class means on coming Tuesday, I will start from restriction endonuclease. And at this point of time, I would like you to ask me questions and if I can answer. So I will be posting the recordings in two parts. This is the first half, and the second half, starting from restriction nucleases, will start on the next Tuesday. And accordingly, I will be posting I've already posted the assignment, but accordingly, I will put the deadline for submission. So I think I see a question for me on the chat. Yeah, the slides are there. The slides are there on the Blackboard. I think I showed that earlier during the class. So let me just show this again. If I can take out the Blackboard, yes. So this is the Blackboard page. Let me start from scratch. This is this course, Molecular Biotechnology. And this is the content folder. If you go to content folder, you have the updated syllabus, my recording for the syllabus, on syllabus. And this is the learning activity for today. So this learning activity will have teaching slides, assignment, and discussion board. And as we are still in the middle of this chapter, so I will put the assignment date accordingly when we finish. So I think I answered the question. Any other questions if you may have? So anybody who came, so I was taking the attendance. Anybody who came later on, could you identify yourself?
Waveney Wallace.
Just a second, just a second.
Just registered yesterday or
Wallace
today.
Waveney?
Yes, Wallace, yes.
Wallace Waveney. So let me just quickly take the attendance.
I may not be there because I just registered. That's
I know,
why.
I saw your name today only. That's fine.
Okay. Okay.
So I did Kaila Aderji, Hauya Anderson, Nafal Bajwa, are you here? Sagin Brunard, are you here? Sagin and Mireke Aisha, Mirenge.
Yes, Professor, I'm here.
You're here. And Melissa Stephenson, are you here? Who is 718908? Could you identify yourself?
That's me. That's Waveney.
Who? Waveney, okay.
Waveney Wallace,
I
yeah.
got you. All right. So Stephenson is, and who is 917412? Hello? Who is 917412? Hello?
Tell them they gotta press *6 to answer you, Professor. To unmute, they have to press *6.
Yeah, the person is unmuted. It's not muted. You see here? 917-412-98 is unmuted. Is not, he or she is not muted. So this will be a problem. Because I admit people while I'm teaching, so I am not able to pause myself. So anyway, so if there are no questions, then I end today's class, and I see you all again on Tuesday.
Okay, thank you.
And I'll be posting the recording later on for the part 1. Okay, so have a good evening and take care. Stay safe.
Thank you.
Professor.
Welcome. Bye.
Okay, thank you.